Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss zymogens. In the previous videos, we discussed how the cell can regulate the catalytic activity of existing enzymes, and that included sequestering enzymes in compartments, availability of cofactors, allosterically regulating the function of the enzyme, as well as covalent modification. And today we'll see there's another way by which the cell can regulate enzymes, and that is by the formation of zymogens. Zymogens are enzymatically inactive precursors of several proteolytic enzymes. So these are enzymes, proteolytic enzymes are enzymes that break down other proteins. The zymogen is converted into an active form by cleavage of some of its covalent bonds. So for example, here is a zymogen, an inactive protease, and by covalently removing some part of this protein, the zymogen becomes an active enzyme, its shape changes and it becomes an active enzyme. Now it's important to note that the conversion of a zymogen into an active enzyme only goes in one direction. It's irreversible, and unlike allosteric and covalent modification, the active enzyme cannot be converted back into the other form, in this case the zymogen form. Let's look at some examples of zymogens to explain this concept. So let's look at this zymogen called pepsinogen. Now pepsinogen is a zymogen that is produced by the chief cells of the stomach. Recall that the parietal cells of the stomach produce hydrochloric acid. The hydrochloric acid in the stomach cavity will break down the pepsinogen zymogen into an active enzyme, proteolytic enzyme called pepsin. And this proteolytic enzyme will now break down your dietary proteins into smaller fragments, smaller pieces. So the formation of zymogens allows the protein to be active only where and when it is necessary. So in case of pepsinogen, the protein needs to act in the stomach cavity. So it's produced by the chief cells, but it's secreted into the stomach cavity. And it's only secreted or becomes active when there is acid present, and that only happens during digestion. So only when acid is present, pepsinogen is secreted, and the pepsinogen is converted by acid into active proteolytic enzyme called pepsin, which will then go ahead and digest your proteins in your food. Another example of zymogens are the blood clotting proteins, several blood clotting proteins, as well as several complements. So examples of zymogens that are blood clotting proteins include prothrombin of factor 2, factor 7, 9, 10, 11, as well as 12. And complement proteins that are zymogens include factor B, D, C1, C2, C3, as well as C4. So once again, these zymogens are in the inactive form, and they only become active when blood clotting is required in case of the clotting proteins, or when there is an immune response that is required in case of complement proteins. So these inactive clotting proteins are made to active clotting proteins when clotting is required. Inactive complement zymogens are made into active complement proteins when an immune response is required. These biological processes such as blood clotting as well as immune response have to be very, very rapid. And the fact that the proteins are already made, they're already synthesized and they are stored. and all the body needs to do is just do a couple of cleavages on the protein to make the protein active. And this really is very important because this saves a lot of time so that blood clotting as well as the immune response can occur immediately. Another example of zymogens are the proteolytic enzymes found in the pancreas or synthesized and secreted by the pancreas. These include proteins such as trypsinogen, chymotrypsinogen, carboxypeptidase, as well as proelastase. And once again, this protein, the zymogens, become active only where and when it is necessary. So in case of this pancreatic zymogens, they will be secreted into the small intestine and they will only get activated during digestion. So these pancreatic zymogens are made by the acinar cells of the pancreas. So they're acinar cells of the pancreas, and then they're secreted through the pancreatic duct. They are made and then they're secreted through this pancreatic duct, and they enter the small intestine where they help in digestion of food. Let us look at an example, chymotrypsinogen activation, as an example to explain this basic concept of activating a zymogen into an active enzyme. So when food enters the small intestine, the duodenum, this triggers the endocrine cells in the small intestine to secrete the hormone called cholecytokinin or CCK. The CCK as well as the vagus nerve will then stimulate the acinar cells to secrete via exocytosis of the zymogen granules that contain all these zymogens such as chymotrypsinogen as well as trypsinogen, and these zymogens will be excreted into the small intestine. Now once the Zymogen chymotrypsinogen is in the small intestine, then another proteolytic enzyme called trypsin will break this peptide bond here between arginine 15 and isoleucine 16. So breaking of this peptide bond will convert the inactive chymotrypsinogen zymogen into pi-chymotrypsin, another form of chymotrypsin where the peptide bond is broken here, and this is called pi-chymotrypsin. Now once the pi-chymotrypsin is formed, it will act on other pi-chymotrypsins and it will remove this part here, this dipeptide, serine 14, arginine 15, and that forms delta chymotrypsin. So that forms delta chymotrypsin. And then it will also remove this fragment here, this dipeptide, threonine 147, 148, and that forms alpha chymotrypsin. And once alpha chymotrypsin is formed, it will have its own unique shape, and this alpha chymotrypsin will now be able to break down the several proteins that you find in your food. So in summary, by forming zymogens, the body makes sure that the enzyme is only active where it is necessary. So in this case, the small intestine, the duodenum, and only active when it is necessary. And so in this case, only during digestion. Another important zymogen that comes from the pancreas is trypsinogen. Now, trypsinogen, again, from the pancreas will enter the small intestine via the pancreatic duct. And once it's in the small intestine, then there is an enzyme called enteropeptidase or enterokinase. This enzyme is found on the surface of intestinal cells. And this enzyme will convert trypsinogen into active trypsin. And as we discussed earlier, the active trypsin will then convert the Zymogen chymotrypsinogen into the active enzyme chymotrypsin. So it's important to keep in mind one of the major reasons why the body makes Zymogens is to make sure that it's only active where it is necessary and is only active whenever it is necessary. So inappropriate activation of Zymogens can lead to various diseases, and probably one of the best examples that we'll talk about is pancreatitis, the inflammation of the pancreas. So pancreatitis is a reversible inflammation of the pancreas. If you look at the pancreas in patients having this disease, we see plenty of inflammatory cells in the affected tissue. This disease is associated with problems in the bile ducts, the biliary tract, and is also related to alcoholism. About 50% of pancreatitis cases have gallstones, and other causes or associations of this disease include certain medications, certain infections, as well as trauma, metabolic disorders, and genetic defects can increase the probability of getting pancreatitis. We still do not know much about the mechanism of the disease, but a lot of findings do show that the Zymogen trypsinogen is somehow activated into active trypsin by enzymes. One of them that have been implicated could be the enzyme lysosomal protease cathepsin B, which can convert trypsinogen into active trypsin. In the disease hereditary pancreatitis, there is mutations in the gene PRSS1, which is the premature cationic trypsinogen activation, and this activation again can lead to pancreatitis. So basically, when the trypsinogen is converted to active trypsin, not only can the active trypsin destroy pancreatic tissue, but this trypsin will also activate other pancreatic zymogens, which also will go on to destroy the pancreatic tissue, and this cellular damage then will lead to the attraction of inflammatory cells, leading to pancreatitis or inflammation of the pancreas. Since trypsin is a very powerful proteolytic enzyme, it's not surprising that our cells also have another mechanism to make sure that in case trypsin becomes activated, we can inhibit the enzyme, and that's by the formation of a trypsin inhibitor. So one example of a trypsin inhibitor is serine protease inhibitor Casal type 1 or SPINK1. So in case trypsin becomes abnormally stimulated or activated, SPINK1 will go and bind to the trypsin and will try to inhibit this active form of trypsin and prevent it from destroying the pancreatic tissue. So therefore, it's not surprising that any mutations in the gene SPINK1 could lead to this loss of function of this inhibitor, and the trypsin activity will be left unchecked, and that will lead to pancreatic damage, and this causes a disease that we call idiopathic pancreatitis. Now, patients with cystic fibrosis also have an increased risk of pancreatitis. So it's quite common to see pancreatitis in patients with cystic fibrosis, and that may be related to the fact that they have decreased water in the ducts, and therefore the mucus that's lining the duct would be quite sticky and would not be able to wash away the zymogens from the pancreatic ducts. So basically these zymogens will stay in the pancreatic duct. It will stay in the pancreatic duct, and if it gets activated, it is going to start destroying the pancreatic tissue, leading to pancreatitis. So once again, we appreciate this very important point that proteolytic enzymes such as trypsin are very, very powerful enzymes, and if they become activated in the organ where they are produced, that can lead to destruction, cellular damage in that organ. And in case of pancreas, that will lead to pancreatitis. So by making zymogens and by other mechanisms, such as by the use of inhibitors, the body tries to restrict the activity of this enzyme only in places where it's needed and whenever it is necessary. So that's a very, very important function of Zymogens. In the next video, we shall look at another way to regulate the activity of existing enzymes, and this is the use of isoenzymes. So until the next video, thank you very, very much for watching and supporting this channel. Have a wonderful day, and hopefully we'll see you in the next video.